Hello everyone. We will continue our discussion in chapter 4. Now we will discuss about the dynamic nature of the plasma membrane. It is apparent from the previous discussion that the lipid bilayer can exist in a relatively fluid state. As a result, a phospholipid can move laterally within the same leaflet with considerable ease. The mobility of individual lipid molecules within the bilayer of the plasma membrane can be directly observed under the microscope by linking the polar heads of the lipids with gold particles or fluorescent compounds. It is estimated that a phospholipid can diffuse from one end of a bacterium to the other end in a second or two. In contrast, the half-life of phospholipid molecule moving across to the other leaflet is measured in hours to days. What you can see in the picture, all the possible motions that a phospholipid can make its flip-flop to the other side of the membrane is most restricted. This finding is not surprising. For a flip-flop to occur, the hydrophilic head group of the lipid must pass through the internal hydrophobic sheet of membrane, which is unfavorable. Flipases are enzymes that actively move certain phospholipid from one leaflet to the other. These enzymes may play a role in establishing liquid asymmetry and may also reverse the slow rate of passive transmembrane movement. The diffusion of membrane proteins after cell fusion. So cell fusion is a technique whereby two different types of cells or cells from two different species can diffuse to produce one cell with a common cytoplasm and a single continuous plasma membrane. Cells are enthused to fuse with one another by making the outer surface of the cell sticky. With that, plasma membranes adhere to one another. Cells can be induced to fuse by addition of certain inactive viruses that attach to the surface membrane, by adding the compound polyethylene glycol, or by a mild electric shock. Cell fusion has played an important role in cell biology and is currently used in an invaluable technique to prepare specific antibodies. In 1970, the first experiment to demonstrate that membrane proteins could move within the plane of the membrane utilize cell fusion were reported by Larry Fry and Michael Edidin of Johns Hopkins University. In their experiment, mouse and human cells were fused, and the location of specific proteins of the plasma membrane was followed once the two membranes had become continuous, to follow the distribution of either the mouse membrane proteins or the human membrane proteins at various times after fusion. Antibodies against one or the other type of protein were prepared and covalently linked to fluorescent dyes. The antibodies against the mouse proteins were complex with dye that fluoresces green, and the antibodies against human proteins with one that fluoresces red. The antibodies were added to fuse cells. They bound to the human or mouse proteins and could be located under a fluorescence microscope. As you can see in the illustration below, the plasma membrane appeared half human and half mouse. That is, the two protein types remained segregated in their own hemisphere. As the time after fusion increased, the membrane proteins were seen to move laterally within the membrane into the opposite hemisphere. By about 40 minutes, each species proteins were uniformly distributed around the entire hybrid cell membrane. If the same experiment was performed at lower temperature, the viscosity of the lipid bilayer increased, and the mobility of the membrane protein decreased, as what is being illustrated in the picture below. These early cell fusion experiments gave the impression that integral membrane proteins were capable of virtually unrestricted movements, as restrictions on protein and lipid mobility. Several techniques allow researchers to follow the movements of molecules in the membranes of living cells using the light microscope. In a technique called fluorescence recovery after photobleaching, integral membrane components in cultured cells are first labeled by linkage to a fluorescent dye. A particular membrane protein can be labeled using a specific probe such as fluorescent antibody. Once labeled, cells are placed under the microscope and irradiated by a sharply focused laser beam that reaches the fluorescent molecules in its path. If the labeled proteins in the membrane are mobile, then the random movements of these molecules should produce a gradual reappearance of fluorescence in the irradiated circle. The rate of fluorescence recovery provides a direct measure of the rate of diffusion of the mobile molecules. The extent of fluorescence recovery provides a measure of a percentage of the labeled molecules that are free to diffuse. Early studies utilizing PROP suggested that membrane proteins move much more slowly in a plasma membrane than they would in a pure lipid bilayer. That a significant fraction of membrane proteins, 30 to 70%, was not free to diffuse back in the irradiated circle. But the PROP technique has its drawbacks. PROP can only follow the average movement of relatively large number of labeled molecules as they diffuse over a relatively large distance. As a result, researchers using PROP cannot distinguish between proteins that are truly immobile and ones that can only diffuse over a limited distance in time allowed. To get around these limitations, alternate techniques have been developed that allow researchers to observe the movement of individual proteins molecules over every short distance and to determine how they might be restrained. In single particle tracking or SPT, individual membrane protein molecules are labeled usually with antibody-coated gold particles approximately 40 nanometer in diameter, and the movement of the labeled molecules are followed by computer-enhanced video microscopy. The result of these studies often depend on the particular protein being investigated. For example, some membrane proteins move randomly throughout the membrane as illustrated in the figure protein A, though generally at rates considerably less than would be measured in artificial lipid bilayer. Some membrane proteins fail to move and are considered to be immobilized as illustrated in the figure protein B, while in some cases, a particular species of protein is found to move in a highly directed manner towards one part of the cell or another as illustrated in the figure protein C. For example, a particular membrane protein may tend to move towards the leading of the trailing edge of a moving cell. It is apparent that plasma membrane proteins are not totally free to drift around randomly on the lipid sea, but instead they are subjected to various influences that affect their mobility. Some membranes are crowded with proteins so that the random movements of one molecule can be impeded by its neighbors as illustrated in the figure protein D. The strongest influence on an integral membrane protein are thought to be exerted from just beneath the membrane on its cytoplasmic phase. The plasma membranes of many cells possess a peripheral network or membrane skeleton, consisting of peripheral proteins situated on the cytoplasmic surface of the membrane. A A certain proportion of a membrane integral protein molecule are either tethered to membrane skeleton or otherwise restricted by it. One approach to studying factors that affect membrane protein mobility is to genetically modify cells so they produce altered membrane proteins. Integral proteins whose cytoplasmic portions have been genetically donated often move greater distance than their intact counterparts, indicating that barriers reside on the cytoplasmic side of the membrane. These findings suggest that the membrane's underlying skeleton forms a network of fences around portions of the membrane, creating compartments that restrict the distance an integral protein can travel as illustrated in protein E. Proteins move across the boundaries from one compartment to another through breaks in the fences. Such openings are though appear and disappear along with dynamic disassembly and reassembly of parts of meshwork. Membrane compartments may keep specific combination of proteins in close enough proximity to facilitate their interaction. Integral proteins lacking that portion would normally project into the extracellular space, typically move at a much faster rate than the wild-type version of the protein. This finding suggests that the movement of a transmembrane protein through the bilayer is slowed by the presence of extracellular materials that can entangle the internal portion of the protein molecule as illustrated in the figure protein F. Membrane lipid mobility. Proteins are huge molecules, so isn't surprising that their movement within the lipid bilayer might be restricted. Phospholipids, by comparison, are small molecules that make up the very fabric on the lipid bilayer. One might expect their movements to be completely unfettered, yet a number of studies have suggested that phospholipid diffusion is also restricted. So, when individual phospholipid molecules of plasma membrane are tagged and followed by high-speed video microscopy, they are seen to be confined for very brief periods and then hop from one confined area to another. This figure shows the path taken by individual phospholipid within plasma membrane over a period of 56 milliseconds. Computer analysis indicates that phospholipid diffuses freely within one compartment, as you can see in the illustration, the one shaded in purple, before it jumps the fins into neighboring compartment, the one shaded in blue, and then over another fins into an adjacent compartment, which is the one shaded in green, and so forth. So, treatment of the membrane with agents that disrupt the underlying membrane skeleton removes some of the fences that restrict phospholipid diffusion. Membrane domains and cell polarity. For the most part, studies of membrane dynamics, such as those discussed above, are carried out on the relatively homogeneous plasma membrane situated at the upper or lower surface of the cell recipient. So most membranes exhibit distinct variation in protein composition and mobility, especially in cells whose very surface display distinct function as illustrated in the figure. In other examples, the receptors for neurotransmitter substances are concentrated in the regions of the plasma membrane located within synapses as seen in the figure. Receptors for low-density lipoproteins are concentrated in the patches of the plasma membrane specialized to facilitate their internalization. Of all the various types of mammalian cells, sperm may have the most highly differentiated structure. A mature sperm can be divided into head, nucleus, and tail, each having its own specialized function. Although divided in a number of distinct parts, a sperm is covered by a continuous plasma membrane, which is revealed by numerous techniques, consists of mosaic of different types of localized domain. For example, when sperm are treated with variety of specific antibodies, as illustrated in the picture, each antibody combines with the surface of the cell in unique topographic pattern that reflects the unique distribution of particular protein antigen within the plasma membrane. Of all the diverse types of membranes, the plasma membrane of the human erectrocyte, or the red blood cell, is the most studied and best understood. There are several reasons for the popularity of this membrane. The cells are inexpensive to obtain, they readily available in huge number from whole blood. They are already present as single cells and need not to be dissociated from a complex tissue. The cells are simply, by comparison with other cell types, lacking nuclear and cytoplasmic membranes that contaminate plasma membrane preparation from other cells. In addition, purified intact erythrocyte plasma membrane can be obtained simply by placing the cell in a dilute salt solution. The cells respond to this osmotic shock by taking up water and swelling, a phenomenon termed hemolysis. As the surface area of each cell increases, the cell becomes leaky and the contents, composed almost totally of dissolved hemoglobin, flow out of the cell, leaving behind the plasma membrane ghost. Fractionation of membrane proteins can be accomplished using polyacrylamide gel electrophoresis or PAGE in the presence of ionic detergent sodium glucosyl sulfate or SDS. So the SDS keeps the integral protein soluble and add a large number of negative charges to the protein with which it associates. Because of the number of charge in SDS molecules per unit weight of protein tends to be relatively constant. The molecules separate from the another according to their molecular weight. The largest proteins move the slowest through the molecular sieve of the gel. The major proteins of the erythrocyte membrane are separated into about a dozen conspicuous bands of SDS and PAGE. Among the proteins are the variety of enzymes that transport proteins and skeletal proteins. Integral proteins of the erythrocyte membrane. So this illustration is a model of erythrocyte plasma membrane showing its major proteins. The most abundant integral protein of this membrane are a pair of carbohydrate containing membrane-spanning proteins called BAN3 and glycoprotein A. BAN3, which gets its name from its position in an electrophoretic gel, is present as a dimer composed of two identical subunits. As blood circulation through the tissues, carbon dioxide becomes dissolved in the fluid of bloodstream and undergoes the following reaction. The peripheral proteins of erythrocyte plasma membrane are located on its internal surface and constitute a fibrillary membrane skeleton, as seen in the figure. So it plays a major role in determining the biconcave shape of erythrocyte and restraining the movement of the membrane's integral protein. The major component of a skeleton is an elongated fibrous protein called spectrin. It is a heterodimer approximately 100 nanometer long, consisting of an alpha and beta subunit that curl around one another. If the peripheral proteins are removed from a rectocyte ghost, the membrane becomes fragmented into small vesicles, indicating that the inner protein network is required to maintain the integrity of the membrane. So, erythrocytes are circulating cells that are squeezed under pressure through microscopic capillaries, whose diameter is considerably less than that of the erythrocytes themselves. So, to traverse this narrow passageway and to do so day after day, the red blood cell must be highly deformable, durable, and capable of withstanding shearing forces that tend to pull it apart. The spectrin action network gives the cell strength, elasticity, and viability necessary to carry out its demanding function. When first discovered, the membrane skeleton of the erythrocyte was thought to be a unique structure suited to the unique shape and mechanical needs of these cell types. However, as other cells were examined, similar types of membrane skeletons containing members of the spectrin and eccrine families have been revealed, indicating that the inner membrane skeletons are widespread. Dystrophin, for example, is a member of the spectrin family that is found in the membrane skeleton of muscle cells. Some mutations in dystrophin are responsible for causing muscular dystrophy, a devastating disease that cripples and kills children. As in the case of cystic fibrosis, the most debilitating mutations are ones that lead to a complete absence of the protein in the cell. The the plasma membranes of muscle cells lacking dystrophin are apparently destroyed as a consequence of mechanical stress exerted on them as the muscle contracts. As a result, the muscle cells die and eventually are no longer replaced.